Now we have the lower extremity. This is likely a lower yield section than the upper extremity. There are fewer types of injuries that you'll be expected to understand, and almost all of them are listed on this page. As we work our way through this chart, let's first just look at all of the nerves. You have the obturator, femoral, common perineal, also known as the common fibular, tibial, and the superior and inferior gluteal nerves. One thing to recognize is that while the nerve roots listed create the first pair and last pair of nerves directly off the spinal cord, the common perineal and tibial are actually terminal branches from the sciatic nerve. Now, let's work our way through each of the nerves listed here. The obturator nerve innervates the muscles along the medial aspect of the thigh, which are all the muscles responsible for adduction of the thigh. It makes sense then that the injuries of the obturator nerve will cause difficulty with thigh adduction and sensory loss of the medial thigh. The next nerve listed is the femoral. This nerve runs anteriorly in the thigh and subsequently innervates the muscles of the anterior thigh. These primarily include the quadriceps muscles, which are responsible for leg extension. It also includes some of the accessory flexors of the hip, namely the sartorius and pectineus muscles. If you lose innervation provided by the femoral nerve, you expect difficulty with thigh flexion, as well as leg extension, and a sensory deficit in the anterior thigh and middle leg. The next two nerves are worth remembering together, in part because they both originate off the sciatic, but also because their motor and sensory attributes are almost entirely mirrored. When you think about the common perineal nerve, also called the common fibular nerve, you should think about the fibula, which lies laterally in the leg. This nerve provides innervation of the muscles of the lateral leg, and also the dorsal surface of the foot. What do you think the muscles of the lateral leg do? They pull the lateral side of the foot out, or evert the foot. When you think about the tibial nerve, you should think about the tibia, which lies medially in the leg. This nerve provides innervation to the muscles of the medial and posterior leg, and also the plantar surface of the foot. What do the muscles of the medial leg do? They pull the medial side of the foot up, or invert the foot. You can use the memory aid PED and TIP to help you remember the functions of each of these nerves respectively. If you look at the motor and sensory deficits for the common perineal and tibial nerves, they match the function paths of the nerves exactly as we've discussed already. The only thing we haven't talked about is the gait deficit that occurs with injuries to the common perineal nerve. When people walk, it's important for them to keep their foot dorsiflexed, or it would either drag along the ground or flap on the ground. This is often referred to as foot drop, and is relatively common in people with multiple sclerosis. The final two nerves we'll look at are the superior gluteal and inferior gluteal. The primary function of the superior gluteal nerve is to abduct the thigh. You can think about it also stabilizing the hip. This is illustrated by the Trendelenburg sign. This is noted when a patient stands on the opposite foot as the nerve injury. When they do, their hip drops because the body can't shift weight onto the leg that it's standing on. Inferior gluteal nerve injuries are different altogether, as it innervates the gluteus maximus. The primary responsibility of this muscle is extension of the hip, which is not a function used explicitly by people on a day-to-day basis. Usually, this injury manifests because deficient gluteus maximus muscles cause difficulty pushing the leg downward. This makes it hard to jump, climb stairs, or even rise from a seated position. Before we move on to the pathology section of the chapter, we just have to review a few more basic concepts relevant to the anatomy and physiology of the musculoskeletal system. One of these topics is the process of muscle contraction and the conduction of electrical signals that leads up to it. These two pictures highlight some of the basic structural components of muscles at a cellular level and at a more structural level. In Figure A, you can see the cellular and receptor level detail of individual myocytes. The invagination that's highlighted represents a T-tubule. On the surface of the cells, you have dihydropyridine receptors, which are voltage-gated calcium channels. Within the cell, lining the sarcoplasmic reticulum, you have ryanodine receptors, which are calcium-induced calcium channels. The sarcoplasmic reticulum and associated channels serve as storage areas for calcium and selective passages for calcium flow, respectively. Calcium, as you may remember from your physiology classes, is integral to the process of muscle contraction. In Picture B, you can see the ultrastructure down to the subcellular microstructure of a muscle fascicle. You can think of muscle fascicles as made of muscle fibers, which are themselves made up of myofibers. Myofibers are made of sarcomeres, which are the smallest functional units of muscle cells. The different bands, zones, and lines detailed in the picture of the sarcomere are worth looking at. In general, the sarcomere extends from Z-line to Z-line. You can remember that the Z is the end of a sarcomere and also the end of the alphabet. Off of the Z-line, actin molecules project in toward the M-line. Off to the M-line, you'll notice thicker lines that represent myosin that is extending out towards the Z-lines. The thin actin fibers and thick myosin fibers overlap always to some degree. Of course, they will overlap more when muscle is contracted, and will overlap less when muscle is relaxed. The M-line is simply the midline of the myosin chains. The H-zone represents the part of the myosin chains that do not overlap with actin. Similarly, the I-band represents the part of the actin chains that do not overlap with myosin. However, if you think about it, these two areas will get smaller with contraction, as the area of overlap between actin and myosin increases. Finally, the A-band is simply the length of the entire myosin chain, from end to end. In summary, the H zone contains myosin only, the I band contains actin only, and the A band contains myosin and the myosin-actin overlap region. The Z line is the site of actin attachment, and the M line is the site of myosin attachment. To remember the distinction, you can use the memory aid HIZ, his shrinkage, and the A band is always the same. If that didn't make sense already, we'll review these concepts as we work through the steps of muscle contraction. The first thing that begins all muscle contraction is an action potential depolarization, which travels down the neuron, opens voltage-gated calcium channels presynaptically, and causes neurotransmitter release. Once the neurotransmitter reaches a postsynaptic neuron, the muscle cell begins depolarizing at the motor end plate. This depolarization will travel down the muscle cell and into the T-tubule pictured in diagram A. This causes depolarization of the voltage-sensitive dihydropyridine receptors, which is also coupled to the ryanodine receptor on the intracellular sarcoplasmic reticulum. This leads to a conformational change that causes calcium to be released from the sarcoplasmic reticulum. That quite literally means calcium was released from the sarcoplasmic reticulum because calcium entered from outside of the cell. This high level of calcium binds to spots on the troponin C molecule. Think C for calcium, which then causes a conformational change, causing tropomyosin to move out of the myosin-binding groove on actin. You can remember that the tropomyosin is the adapting molecule between the calcium-binding troponin and the contraction-generating myosin portion by keeping in mind that the name incorporates both sides. Myosin then releases prior-bound ADP molecules and is displaced on the actin filament, the so-called power stroke, which is actually how myosin molecules move along actin. We'll look at this concept in more detail on the next page. Before we do that, though, we're going to discuss the different types of muscle fibers. This may seem a little tricky at first, but it's actually just a quick fact to tuck away. Basically, there are two types, type 1 and type 2. You should think about type 1 fibers as being responsible for sustained contraction, while type 2 fibers are responsible for fast twitch. Type 1 fibers are slow twitch, and you can remember everything you need to know about them with the memory aid, one slow red ox. That is, they are slow twitch, they are red fibers, meaning that they have lots of mitochondria and lots of myoglobin, and OX, which means they utilize full oxidative phosphorylation in their mitochondria. Slow twitch muscles tend to be postural muscles. For example, your paraspinal muscles. These muscles have long sustained contractions. Just think about when you're walking or just sitting up straight. This allows the ample mitochondria to have the time to go through oxidative phosphorylation and create enough ATP to continually power these muscles. In contrast, type 2 fast-twitch muscles are white because they do not have a lot of mitochondria or myoglobin. They primarily use anaerobic glycolysis. If you think about it, though, this is probably for the best. Fast-twitch muscles include muscles like your biceps or your extraocular muscles. These muscles can't wait for additional ATP to be generated through oxidative phosphorylation. They need ATP fast. This muscle is also the type of muscle that commonly hypertrophies in weight training. As promised, we've now returned to the topic of muscle contraction. Before we were looking at the sarcomere and its greater organizational structure within muscle fibers and even muscle fascicles. Here, we'll look within the sarcomere to see why the areas of actin-myosin overlap that we discussed earlier actually contract. The basic idea is that heads from the myosin chains attach to actin and stroke along them, a process that's dependent on ATP. You can think about the first step as just having a myosin head that's ready to attach to actin. In the resting state, that myosin head can't attach because tropomyosin blocks binding. However, when the signal for muscle contraction comes, calcium rushes into the cell and binds to troponin C, causing the tropomyosin to move out of the way and allow binding of actin to myosin. The myosin is presumably already in the cock position, so the next step is for the myosin head to power stroke along the actin, which results in the loss of a phosphate group. Subsequently, the ADP falls off the myosin head as well. This is what triggers the power stroke, causing the actin and myosin fibers to slide past one another and causing contraction on the larger scale. Soon, an ATP molecule will rebind the myosin molecule and cause it to fall off the actin molecule. Think of the T in ATP as standing for terminate, as in it terminates the connection between actin and myosin. The energy from the ATP is then used to reset the myosin head into the cock position, leaving behind an ADP molecule on the myosin head. Repetitions of this cycle lead to repeated crossbridge formation and shortening of the sarcomeres. Now, it would be convenient if all the muscles contracted the same way, but unfortunately that's not the case. The muscle contraction that we've discussed so far primarily describes skeletal and cardiac muscle. Smooth muscle, however, contracts using a different system. The precise mechanism for smooth muscle contraction is probably lower yield than the mechanism for striated, but if you have the time, it's worth learning for completeness' sake. Focus on the major differences between the two. The primary difference is that the smooth muscle does not employ the troponin-tropomyosin system the same way. That said, the first few steps are the same. Action potential is fire, causing smooth muscle membrane depolarization, causing opening of voltage-gated calcium channels. This is where it changes though. Rather than binding to troponin C, the calcium binds to calmodulin. This calmodulin complex then activates the myosin light chain kinase, which, as you can likely figure from the name, will phosphorylate myosin chains. When myosin is phosphorylated, it will form cross bridges and cause contraction of muscle as it moves along the actin molecule. The molecule that counters the effect of MLCK is of course myosin light chain phosphatase, which dephosphorylates the myosin molecule and causes relaxation. You can think about smooth muscle contraction as a battle between the kinase and the phosphatase. In the presence of calcium-activated calmodulin, the kinase efforts win and cause contraction. Interestingly, calcium is the primary signal for muscle contraction, but there are other signals that cause muscle relaxation. One of the most important ones is nitric oxide. This can be formed endogenously in cells like endothelial cells, which can lead to blood vessel smooth muscle relaxation. This, of course, leads to vasodilation. The nitric oxide activates the enzyme guanylate cyclase, which increases levels of cGMP. These increased cGMP levels cause inhibition of MLCK. This means that MLCP-mediated relaxation will win out over MLCK-mediated contraction. Now we've talked a considerable amount about the physiology of muscle, which admittedly is pretty complicated. But before we turn our attention to the pathology section, we have one more topic to cover, and that's bone formation. There are two processes of bone formation: endochondral ossification and membranous ossification. The difference between the two is relatively straightforward. With endochondral ossification, cartilage is laid down as a precursor for bone growth. It's sort of like making an outline for an essay. After making the outline, you have a structure already that you can follow for writing the first draft. After you make the first draft, you can go over it one last time and make a final draft. That's basically how endochondral ossification works. You start with a cartilage model, or the outline. You then convert it into woven bone, or a first draft. While woven bone is actually bone, its fibers are fairly haphazard and lack strength. However, woven bone is then converted into lamellar bone, or the final draft, which is more structurally and mechanically sound. Endochondral ossification is the method of formation of all long bones in the body, bones like the femur and the humerus. Extending the metaphor, it takes a lot of drafts to make a long essay, just like it takes several formational steps, endochondral ossification, to make long bones. The second process, membranous ossification, is not entirely dissimilar. It's basically writing the first draft without the outline. There's no cartilage model in membranous ossification. There's only a first draft written from scratch, or woven bone laid down without a cartilage framework. Then the woven bone is refined into lamellar bone, or a final draft. This process is not used for long bones, but rather for flat bones, which includes bones like the skull, or the facial bones, which, as a side note, are the major sites for active, erythrocyte-producing red marrow later in life.